Big data has become a bit of a buzzword recently, and I think that essentially the definition of big data is that we are now able to collect data at a much higher rate than we used to be able to do. And what that does is it generates data sets which are much, much larger than we're normally used to dealing with. And what impact this has is several-fold. The first is that it actually gives researchers new opportunities to actually mine data sets that were not available before. But what that also means is that we've now got to learn new tools that are going to let us mine those data sets. But I think also very, very importantly, we've got to come at those large data sets with a very clear hypothesis in mind that we want to test. So it's not simply, oh, I'm going to go to that data set which contains information on 10,000 samples and I'm going to look at it. What you have to do is you have to go to that large data set with a hypothesis in mind which you want to test against the data set. And that's quite different than the way things used to work, which was you have a hypothesis and then you generate an experiment to test that hypothesis. Now we can actually leap over some of that and go straight to the data set to test hypotheses. So there are many. So I guess the first kind of classic large data set that people would be most familiar with is DNA sequence. So the genome sequences were coming out, Arabidopsis thaliana in 2000, the human genome, and then we've had many, many other genomes coming out since then at an ever increasing rate due to new technologies, particularly next generation sequencing, now giving us an Arabidopsis thaliana genome in less than a day. So that's probably the one that everyone thinks about when thinking about large data sets. But there are many other types of instrumentation that will generate large data sets. So for example, proteomic data sets. So this is generated by new advances in mass spectroscopy. You've got metabolomic data sets, so all the metabolites in an organism. You've got transcript data sets, so all the expressed genes in an organism. Stuff that I work on is the ionome, so all the elements that accumulate in an organism. So I think that as instrumentation develops, it allows us to measure more things in more samples more quickly and also cheaper. So that's quite key. So as we start to approach these large data sets, then what you very, very quickly realize is that you want to essentially look at those data sets. You want to test a hypothesis. You want to somehow take that data set and reduce it in some way so that you can get to the stuff that you're interested in. So how do you do that? When you start to think about these large data sets, what tools do you use to manipulate these data sets? You can't just simply print it out on a piece of paper and then just go through the list. You've got 100,000 samples there and you've measured each one for 10,000 SNPs. This is not possible to do by hand anymore. So you've obviously got to use a computer somehow. So how do you do that? Well, the first thing that comes to mind, well, let's just import it into Excel, right? Well, the trouble with that is that Excel has a certain maximum amount of rows, and these large data sets just can't go into Excel. They very rapidly overwhelm something like Excel. So very, very quickly, you realize that if you actually want to manipulate these large data sets, then you've got to write computer programs. Now, these aren't complicated computer programs. These are fairly simple scripts to do relatively straightforward tasks, like change columns to rows, take the means, the averages, these kinds of things, extract certain columns and put them into a new file, these kinds of file manipulations. So they require relatively simple scripting, but without that scripting, what it means is you have to rely on bioinformaticians to do a lot of this work, which if you just learn a little bit of computer programming, then you could actually do yourself and you'd be much, much more efficient and independent. The big opportunities. Well, I think for me personally, and this is obviously a personal opinion, the big opportunities in plant science research using big data sets for me is what I would describe as ecological genomics. So I think that we've made tremendous strides in plant research using what people like to call genetic model systems like Arabidopsis thaliana, in which we work in the lab, we generally delete genes, we try to understand what those genes do. But what we're starting to realize is that what you learn about deleting genes in the lab isn't necessarily the same as what those genes might do in the environment. And more particularly, when you study one particular genetic model, then what you're only looking at is a small snapshot of the genetic variation that is actually in that species. And so you're missing huge amounts of information. So I think one of the big opportunities now is actually translating what we're starting to learn about using molecular genetic tools into the ecological function of genes. And I think one of the big upsides of that is as we start to understand what the ecological function of genes is, that's going to very, very strongly feed back into how do we then adapt our crops to particular ecological situations like high salinity, drought, etc. And I think that's obviously going to become more and more important as we're facing changing climate, and the current crops that we have are just not going to be adapted to early frosts, late rains, high winds, can't get the tractors in the fields, we've got the wrong varieties in the fields, etc. So I think that's where I think there's a big opportunity. And I think there's still a lot of space in the UK. We've got a lot of excellent scientists working on a lot of really interesting ecological problems. And I think if we could bring in genomics and fuse these two areas together, I think we've got a tremendous world-beating set of opportunities.